Good morning, everyone. Today, we are honored to present our research entitled From Local Leaves to Solar Energy, Natural Dye Potential of Bangun-bangun, Andaliman, and Cassava Leaves in Dye-Sensitized Solar Cells, DSSC, Applications. This research is a collaborative effort by a dedicated team of students from Insert School Name, including Omar, Ghani, Mutyara, Shafin, Mubarak, Akbar, Hasibuan, Rasyid, Bakhtiar, Sibarani, Alia, Kanaya, Tarigan, Kanza, Alya, Rafiqa, Mohammed Rehan, Armando Pohan, and Mohammed Fidel Ghani. With the supervising teacher Alia Ahmatul, Umri, Hasibuan. We are all students from Shafin Alia Lower Secondary. Our study explores the innovative use of natural dyes extracted from local North Sumatran plants as sustainable photosensitizers in solar cells. By harnessing the unique pigments of bangun-bangun, Andaliman, and cassava leaves, we aim to provide an eco-friendly, cost-effective, and culturally grounded solution for renewable energy. Through this presentation, we will guide you through the background, objectives, methodology, and potential impact of our research, demonstrating how local biodiversity can contribute to both science and society. The increasing demand for renewable energy worldwide has become a critical issue as fossil fuel resources continue to decline and environmental pollution escalates. Among the various renewable energy technologies, solar energy stands out as a sustainable, clean, and widely accessible option. In particular, dye-sensitized solar cells, DSSCs, have gained attention due to their cost-effectiveness, flexibility, and environmentally friendly nature. Unlike traditional silicon-based solar cells, DSSCs rely on photosensitizer dyes to capture sunlight and convert it into electrical energy. However, the majority of DSSCs use synthetic dyes, which are expensive, toxic, and non-biodegradable, leading to environmental hazards and limiting their accessibility, especially in rural or remote areas. To address these challenges, our research focuses on the potential of local North Sumatran plants as sources of natural dyes for DSSC applications. Bangun-bangun, Coleus amboinicus, contains anthocyanins, producing red or purple pigments. Andaliman, Zanthoxylum acanthopodium, contains flavonoids, producing orange-red pigments, and cassava leaves, Manihot esculenta, are rich in chlorophyll, producing green pigments. These natural dyes are not only abundant and renewable but also biodegradable, non-toxic, and safe for handling. Utilizing them in DSSCs provides an environmentally responsible and cost-effective alternative to synthetic dyes. This research is significant because it integrates sustainability, local biodiversity, and renewable energy technology. By leveraging plants readily available in North Sumatra, this study promotes the sustainable use of natural resources while providing opportunities for scientific exploration and STEM-based learning. Furthermore, by demonstrating the feasibility of natural dye-based DSSCs, this research encourages community involvement, renewable energy awareness, and educational engagement in eco-friendly technologies. Despite the benefits of natural dyes, several challenges remain. Synthetic dyes still dominate DSSC production due to their high efficiency, and limited studies have systematically investigated the combination of local North Sumatran plants for solar energy applications. Therefore, this research aims to answer the question, can natural dyes extracted from bangun-bangun, Andaliman, and cassava leaves serve as effective, sustainable photosensitizers in DSSCs while promoting renewable energy awareness and local resource utilization? The objectives of this research are to extract natural pigments from bangun-bangun, Andaliman, and cassava leaves in a safe and non-toxic manner. To fabricate DSSC photoanodes using TO2 and apply the natural dyes as photosensitizers. To evaluate the efficiency and stability of DSSCs using these natural dyes under simulated sunlight. To promote renewable energy knowledge, sustainable practices, and community awareness. Through this study, we aim to demonstrate the scientific, environmental, and social potential of natural dye-based DSSCs environmentally. Using biodegradable plant-based dyes reduces chemical waste and pollution. Scientifically, this study explores the efficiency and feasibility of local natural dyes in energy applications. Socially and economically, it encourages local communities to cultivate and utilize indigenous plants, supports STEM learning, and inspires engagement with sustainable energy solutions. Dye-sensitized solar cells, commonly referred to as DSSCs, have emerged as a promising technology in the field of renewable energy. Unlike traditional silicon-based solar cells, which require high-cost materials and complex manufacturing processes, DSSCs rely on a photosensitizer dye to absorb sunlight and convert it into electricity. The performance of a DSSC largely depends on the efficiency of the dye in capturing light and facilitating electron transfer. While synthetic dyes, such as ruthenium-based compounds, have been widely used due to their high efficiency, they are expensive, non-biodegradable, and potentially hazardous to the environment. Natural dyes derived from plants offer a sustainable, cost-effective, and eco-friendly alternative. These dyes are abundant, biodegradable, and safe for handling, making them particularly suitable for educational and research purposes. Studies have shown that pigments such as anthocyanins, flavonoids, and chlorophyll exhibit strong light absorption in different regions of the visible spectrum, which is crucial for enhancing DSSC performance. Anthocyanins, found in plants like bangun-bangun, absorb red to purple wavelengths, providing effective energy conversion. Flavonoids, such as those present in Andaliman, absorb in the orange-red spectrum and act as natural antioxidants, enhancing electron stability. Chlorophyll, abundant in cassava leaves, absorbs both blue and red light, complementing the other pigments to maximize sunlight capture. Previous research has demonstrated that combining multiple natural dyes can improve DSSC efficiency by broadening the absorption spectrum and facilitating better electron transfer. For example, Kusumawati et al. 2024 reported that synergistic combinations of plant-based dyes significantly enhanced solar cell performance compared to single dye systems. These findings suggest that the integration of multiple local plant pigments, such as bangun-bangun, Andaliman, and cassava leaves, could offer an innovative approach to creating efficient and sustainable DSSCs. Beyond technical considerations, the use of local plants in DSSCs also promotes community engagement and sustainability. By sourcing dyes from plants native to North Sumatra, this approach encourages the cultivation and sustainable utilization of indigenous flora. It also provides opportunities for students and researchers to learn about renewable energy, natural dye chemistry, and environmental stewardship. In this way, natural dye-based DSSCs not only contribute to scientific advancement but also foster social, educational, and environmental awareness. In conclusion, natural dyes from bangun-bangun, Andaliman, and cassava leaves hold significant potential as photosensitizers in DSSCs. They provide an environmentally friendly, cost-effective, and accessible alternative to synthetic dyes, while also promoting renewable energy education and community engagement. This study aims to build on existing research by exploring the combined use of these local plant pigments in DSSCs, with the goal of developing a sustainable, practical, and socially impactful solar energy technology. This research applies a research and development approach to explore the potential of natural dyes from local North Sumatran plants as photosensitizers in dye-sensitized solar cells. The study combines experimental procedures, chemical analysis, and community engagement to ensure both scientific rigor and practical applicability. The first step involves the careful selection and harvesting of bangun-bangun, Coleus amboinicus, from Caro, Andaliman, Zanthoxylum acanthopodium, from Caro, and cassava leaves, Manihot esculenta, from Simalungan. The leaves are thoroughly washed to remove dirt and impurities. Pigments are then extracted using safe solvents such as distilled water or ethanol. Extraction methods include maceration and soxhlet techniques, chosen to maximize pigment yield while maintaining environmental safety and minimizing chemical hazards. The resulting extracts are filtered and stored in dark containers to prevent degradation and ensure their stability as natural dyes. Next, the photoanodes of the DSSCs are prepared by coating conductive glass substrates with titanium dioxide (TO2) paste. The TO2 layer is sintered to create a porous surface, which allows for maximum absorption of the natural dyes. Each leaf extract is applied individually and in combinations to the TO2-coated electrodes, enabling a comparative evaluation of single versus mixed dye systems. This step is critical for understanding how the pigment's absorption spectra interact to influence overall solar energy conversion efficiency. The assembly of the DSSC involves pairing the dyed photoanode with a counter-electrode and introducing a suitable electrolyte solution to facilitate electron transfer. Careful sealing ensures stability and prevents leakage during testing. The fully assembled DSSCs are then evaluated under simulated sunlight to measure key performance indicators, including open circuit voltage, short circuit current density, fill factor, and overall energy conversion efficiency. These measurements provide a quantitative assessment of how well the natural dyes function as photosensitizers in real-world conditions. In addition to laboratory experiments, this research emphasizes educational and community engagement. Workshops are conducted with students and local communities to demonstrate the extraction of natural dyes and the fabrication of DSSCs. This not only fosters STEM learning but also encourages sustainable practices, such as planting and cultivating dye-producing plants. Furthermore, collaborations with local farmers ensure a reliable and sustainable supply of raw materials while promoting economic empowerment through the cultivation of bangun-bangun, Andaliman, and cassava leaves. Finally, the collected data is analyzed using descriptive statistics and comparative techniques to determine the most effective dye combinations and their corresponding solar cell performance. The study also evaluates the practical feasibility of implementing these natural dye-based DSSCs in educational and community contexts. By combining scientific experimentation, sustainable resource utilization, and community engagement, this methodology provides a holistic framework for developing eco-friendly, cost-effective, and locally sourced solar energy solutions. The expected outcomes include functional DSSCs using natural dyes, enhanced awareness of renewable energy, and active participation from students and community members in sustainable scientific practices. In conclusion, this research demonstrates the significant potential of local North Sumatran plants bangun-bangun, Andaliman, and cassava leaves as natural dyes for dye-sensitized solar cells. By harnessing these pigments, we provide an eco-friendly, cost-effective, and sustainable alternative to synthetic dyes, reducing environmental pollution while promoting renewable energy accessibility. The integration of natural dye extraction, DSSC fabrication, and community engagement highlights not only the scientific value of this study but also its educational, social, and economic impact. Through this work, we hope to inspire the use of local biodiversity and innovative technologies, encourage sustainable practices, and foster awareness of renewable energy among students, researchers, and communities. Ultimately, this research contributes to a vision of green, accessible, and community-centered energy solutions, demonstrating that scientific innovation and environmental stewardship can go hand in hand. Thank you.